This session is about the nucleus, about the nucleolus. But before beginning about the nucleolus, I'll give you a brief introduction about the nucleus, just only a brief introduction. The nucleus. Nucleus is the first organelle in the cell which was to be discovered, and nucleus is considered to be the storage of genetic information, and it is considered to be the cell's control center because the genetic material is present inside the nucleus. Nucleus is considered to be the cell's control center, that it contains the genetic material in the form of either RNA or DNA. And the nucleus is double walled, or it is a membrane bound organelle. Nucleus is one of the membrane bound organelle which is found inside the cell, and it is double walled. The nuclei, there are cells which most of the cells have a single nucleus, but there are also cells which have multiple nuclei, and even cells which lack nuclei. The cells which lack nuclei, the example is RBCs. We know that RBCs are the cells which do not have a nuclei, and that is because they have those cells, or RBCs can carry as much as amount of hemoglobin, and that's the reason why they don't have a nuclei. So most of the cells like nerve cell, cardiac cell, muscle cell, all cells have nucleus, that too a single nucleus, and very few are multinucleated condition. And the genetic material, the amount of genetic material also varies from individual to individual, or from organism to organism. So this is a picture of a nucleus. You can see here inside the cell. Inside the cell, usually you can see the common structure of the cell which all of you might have learned, and inside the cell you can see a nucleus, and inside the nucleus there is genetic material. So this is a nucleus. You can see a nucleus. Nucleus is a double walled structure with nuclear pores. That means there are openings in the nucleus. This is about nucleus. I'm talking about nucleus. Nucleus is a double walled structure. They have a double walled membrane with openings in between, and these openings are called the nuclear pores. And the function of this nuclear pore is to transfer the RNA or the proteins to and from the nucleus to the cytoplasm. So here you can see the nucleoplasm. This is a nucleus with double walled membrane. See, it's a double walled structure, double walled membrane it have, and inside the nucleus there is chromatin. Inside the nucleus there is chromatin and nucleoplasm or nuclear matrix. The mesh-like structure which is present inside the nucleus is called the nuclear matrix, and inside the nucleus you can see the nucleolus. So nucleolus is the structure which is found inside the nucleus of a cell. Then, so the main functions, this is a structure of a nucleus in detail. So nucleus, I said, it is the control center of the cell. So inside the nucleus there are, you can see the nucleolus. This is the nucleolus. On one side or one part of the nucleus you can see the nucleolus, and nucleus have, this is nucleus. Nucleus have large number of openings called the nuclear pores, and inside the nucleus there are chromosomes. Chromosomes are like this, will be located like this, and nucleolus is like this. So it helps in regulating the gene expression of the cell, and the nucleus inside the nucleus synthesis of RNA takes place, and the RNA will move out of the nucleus through the nuclear pores. Then the nucleus, based on the structure of nucleus, the cells are of two types, prokaryotes and eukaryotes. Prokaryotes are those cells which don't have a true nucleus. Prokaryotes and eukaryotes, these are the basic difference between two types of cells, prokaryotes and eukaryotes. Prokaryotes and eukaryotes. Prokaryotes don't have a membrane-bound cell, membrane-bound nucleus, but eukaryotes have a true Nucleus, that means the nucleus with membrane. So eukaryotic cells have a double-walled nuclear membrane, and on the nuclear membrane there are nuclear pores. Inside the nucleus there is the nucleolus. Then, coming to the eukaryotes and prokaryotes. Eukaryotes, there is no distinct nucleus. So in eukaryotes, prokaryotes don't have a prokaryotes example bacteria. The prokaryotes example is bacteria. Bacteria don't have a nuclear membrane. So the DNA will be present inside, or the genetic material will be present inside the cytoplasm itself. So the synthesis of RNA and protein takes place in the cytoplasm itself. That takes place in the case of prokaryotes. So prokaryotes are those cells which don't have a true nucleus, because there is no true nucleus. That is because the nucleus is not having a double-walled membrane. The synthesis of RNA and protein takes place in the cytoplasm itself. But in the case of eukaryotes, we are talking about the eukaryotes means true nucleus. Eukaryotes are those cells which have a true nucleus. So this eukaryotic cells, yes, see, this is the nucleus. This is the nucleus and this is the cytoplasm of the cell. So in eukaryotes, the cell will be having cytoplasm, and inside the cytoplasm there will be nucleus. Inside the nucleus you can see the genetic material, that is, it can be either DNA or RNA. So here I have given the example of a DNA, that is, inside the nucleus DNA is present, and from the DNA, as a result of transcription, RNA will be formed. We will study the details of transcription etc. later, but just remember here, inside the nucleus, from DNA, RNA is synthesized, and this RNA is exported, or this RNA moves out of the nucleus to the cytoplasm. See, this is the cytoplasm. So what happens? The synthesis of RNA takes place inside the nucleus, and the RNA will move out of the nucleus through the nuclear pores to the cytoplasm, and the synthesis of proteins takes place where? In the cytoplasm. That is what. In eukaryotes, the synthesis of RNA takes place inside the nucleus. From the nucleus, the RNA moves out of the nuclear pores to the cytoplasm. And in the cytoplasm, what happens? The protein synthesis takes place. But in prokaryotes, what is happening? Prokaryotes don't have a true nucleus. That means no nuclear membrane. So, the synthesis of RNA and protein takes place in the cytoplasm itself. So these are the, we'll just have a brief look only. These are the nuclear pores in the nucleus. See, this is the nucleus, and through this nuclear pores only, the RNA will move to the cytoplasm. For to and fro movement of proteins and RNA takes place through this nuclear openings called the nuclear pores. Nucleus is having a nuclear membrane, and from the nuclear membrane is continuous with the endoplasmic reticulum, and the nuclear membrane contains so many membrane channels. Then nuclear membrane is supported by nuclear lamina, which gives strength or support to the nuclear membrane. This is the nucleolus. So let's see in detail what nucleolus is. Inside the nucleus, you can see here, this is the nuclear envelope. So, nucleolus is a non-membrane bound structure. See, this is a nucleolus. This is nucleolus. Nucleolus is the structure which is found inside the nucleus. Where is it found? It is found inside the nucleus. But what about its structure? It's not a membrane bound organelle. That is important. It is a non-membrane bound structure. So, nucleolus is a non-membrane bound structure that is found inside the nucleus. Clear? Nucleolus, where is it found? Inside the nucleus. Well, how about the structure? It is a non-membrane bound structure. Then, it is composed of proteins and nucleic acids. The nucleolus is composed of proteins and nucleic acids such as RNA. It is composed of proteins or composed of nucleic acids such as RNA. Mostly it is RNA and only very less amount of DNA. That is what is nucleolus. So, nucleolus is a non-membrane bound structure found inside the nucleus. It is composed of what? Proteins and nucleic acids, mainly RNA and very less amounts of DNA. And the main part of the cell, that is nucleolus, produces ribosomes. The function or done by this nucleolus is actually the processing and ribosome assembly. Processing and ribosome assembly is the function done by the nucleolus. See, it has a network-like structure. It is not a membrane-bound structure. So, the nucleolus, the ribosomal RNA, the nucleolus is mainly involved in the ribosomal rRNA. They are mainly involved in the rRNA transcription and processing of the ribosome assembly, rRNA synthesis. They are mainly involved in the rRNA synthesis, and so they serve as the ribosome production factory, and this nucleolus is called ribosome production factory. Ribosome production factory. These are the important things which has to be. It is called the ribosome production factory. So, nucleolus, they are mainly involved in the synthesis of rRNA. And coming to the structural details, already I've told it does not have a membrane, but it has three main parts. The nucleolus has three main parts, and the three main parts are the fibrillar center, the dense fibrillar component, and the granular components. They have three regions, that is the fibrillar center, the dense fibrillar component, and the granular component. Actually, and these three regions, or all the three regions, the fibrillar, the dense fibrillar, and the granular components, are involved in the rRNA transcription, processing, and the ribosome assembly. Now, the nucleolar organizer regions, or NOR, are the chromosomal regions which is responsible for the formation of the nucleolus. NORs, or nucleolar organizer regions, are responsible for the formation of the nucleolus. And in human beings, the NORs, in humans, the NORs are located on the short arms of the acrocentric chromosome. We'll see what acrocentric chromosome is. So they are the acrocentric chromosomes like chromosome 13, 14, 15, 21 and 22. These are the chromosomes in which the NOR, or the nucleolus organizer regions, are located. Nuclear organizer regions are located in this chromosome 13, 14, 15, 21 and 22. And these regions code for the 5.8S RNA, 18.S rRNA, and 28S rRNA. They mainly code for these rRNAs, and they are found between the repetitive heterochromatic DNA sequences and the telomeres. They are found between the, like this is the, they are found between the telomere and the repetitive sequences. Almost this will be the region of the NORs, or the nucleolar organizer region. The part of the chromosome which is responsible for the organization of the chromosome of the nucleolus is called NORs. They are located in 13, 14, 15, 21 and 22 chromosomes. Now, let's see the structure of a chromosome. So, chromosome, usually the structure which is mainly there are different stages in the life cycle of a cell, and that too in particular to study the structure of the chromosome. The metaphase and the prometaphase is taken into consideration because at that time the chromosome is found at its maximum condensed state. So this is an unreplicated chromosome, and as a result of DNA synthesis and condensation, I said the metaphase and the prometaphase are usually considered for studying the structure of chromosome because the chromosomes are found in a condensed stage at that stage of the cell cycle. So in the DNA, just I'm showing this picture just to give you an idea about certain terms which is used in the chromosome and that we have already mentioned in the human genetics section also. These are the two sister chromatids. So this is the chromosome will be found in a coiled manner, and during metaphase the chromosome will take a structure almost like this, and these are called the sister chromatids. Two sister chromatids, and the sister chromatids will be joined at a particular region called the centromere. This is very important. The two sister chromatids are joined together at a particular region, and that region is called the centromere. So centromere, and adjacent to the centromere there is a structure called kinetochore. And now based on the position of centromere, so centromere, the mitotic chromosome will have a constriction at the middle portion, and that region is called a centromere. So centromere is a constricted chromosomal region at which the two identical sister chromatids are attached. And the centromere is very important during mitosis or during cell division. Based on the position of the centromere or this constriction, chromosomes are of different types. Metacentric chromosome, submetacentric chromosome, acrocentric chromosome, and telocentric chromosome. So, metacentric chromosome. In metacentric chromosomes, the chromosome will be located at the center of the chromosome. That is known as metacentric chromosome. So, if we draw like this, the metacentric chromosome will be like this. This is the centromere. This is the centromere. If it is in the bent structure, you can see like this. That is metacentric chromosome located at the center of the chromosome. So these are the two arms. Now, if the chromosome or if the centromere is located a little bit away from the center, that's known as submetacentric chromosome. Submetacentric chromosomes. Then very submetacentric chromosome, that is which is much more far from the center, that is very metacentric, submetacentric chromosomes. Then acrocentric chromosome. Acrocentric chromosome means the centromere will be almost towards the tip of the chromosome, that is acrocentric chromosome. Now the next type is telocentric chromosome. Telocentric chromosome. Telocentric chromosome is a type of chromosome in which the centromere is located towards the tip. Actually, that is that is telocentric chromosomes. Actually, it's very rare in existence. Telocentric chromosomes. This is the chromosome will be, the centromere will be towards the tip of the chromosome, that is known as telocentric chromosome. So in human beings there are different types of chromosome. Here I've written the chromosome number 1, chromosome number 2. The different chromosomes in our body is of different types. Some are metacentric. We have 23 pairs of chromosomes, and out of that some are metacentric, some are submetacentric, some are telocentric, some are acrocentric, and some are telocentric, like that. This is about the centromere. So centromere is the middle region, or it is the construction of the chromosomes, and associated with the during mitosis the centromere gets attached to the mitotic spindles, and the sister chromatids will separate and move towards the opposite poles. Then next is telomere. Telomere is called the centromere is the central portion of the chromosome, but telomere is the tip of the chromosome. In this picture I have shown here in pink color, you can see the chromosome tip. In this picture you can see the chromosomes are almost X-shaped. So the tip of the chromosome is given in pink color. So this is known as the tip of the chromosome, and telomere is the tip of the chromosome, or the DNA sequences that are seen at the end of the eukaryotic chromosome are called telomeres. So this is the chromosome. This picture you can see the pink structures. This is the chromosome. This region. This is the tip of the chromosome. These are the tip of the chromosome. So the DNA sequences that are found at the ends of the eukaryotic chromosomes is called the telomere. Telomere is the region which is found at the end of the chromosome, and the sequences and this telomeres, they are called the cap because they are found at the end of the chromosome. They are called the cap of chromosomes, or they are found at the end of the chromosome, and the telomeres are required for the stability and replication of the chromosomes. And these telomeres are heterochromatic and they are highly condensed, and they are often seen associated with the nuclear envelope too. And these telomeres are comprised of a repeated sequence. So telomeres are found at the end of the chromosome. They are also called the tip of the chromosome because they are found at the tip of the chromosome. The wear and tear which happen to the nucleus, which happen to the chromosome during cell division, is more at the end. So when a person is, it's easy to remove a bead from one side of a string rather than removing a bead. If you have just for example, just imagine if you have a string of beads, it's easy to remove the string which is at the tip of the string, but it is difficult to remove the bead from the center of the string. That is what. So these telomeres are actually the beads which are at the end of the string. So they are very much susceptible to the wear and tear when a cell undergoes division, or when the nucleus undergoes, chromosome undergoes division. And coming to the details of the telomeres have highly repeated DNA sequences. They are composed of repeated sequences called the TTAGGG, and these are called the repeated sequences or tandem repeats. In mammals, the telomere DNA, or the DNA at the tip of the chromosome, have a tandem repeated sequence like TTAGGG, TTAGGG, like that. And it is followed by a 3' rich single-stranded overhangs. And they take the telomere DNA adopt a T-loop structure where the tip of the end fold back inside to form a 3' overhang inwards. That is, if this is the tip of the chromosome, just imagine this is the tip of the chromosome, and this tip will be having, for elaborated view, if you take this tip, will be having the repeated DNA sequence, and they will take a loop. Why this loop? Actually, this loop is taken by the chromosome ends to minimize the chromosome loss during the replication. So that is very important. And the telomere is very much associated with the aging also. Telomeric DNA is almost same in all humans, and that is other vertebrates also. And more number of times a cell divide, as I said, telomere is the bead at the end of a string, so it is more susceptible to wear and tear. So as as the age advances, or as the number of divisions of the cell increases, what happens is the telomere will be, or the tip of the chromosome will be lost slightly. That is why it is given here. The length of the telomere ranges from 3000 base pairs as low as 1,500 in elderly people. The number of base pairs in the end of the chromosome will be less compared to the number of chromosomes at the young stage. See, here you can see the chromosome of this is a chromosome of a baby or a child, but as the age advances, what happens? The telomere will be shortened. And that is the reason why the muscle tone lessens. Many of this, like the tonicity of muscles and the activity of the individuals vary between an adult and a young. And the telomere importance of one advantage is the telomeres will not shorten in case of cells in heart muscles. Heart muscles, it will not get shortened. And also, they are not shortened in case of cancerous cells too. That is why they undergo fast replication. So telomere length will not be shortened in case of heart muscles, as well as it is not shortened in case of cancer cells. That is how and actually this invention of this cancerous cells was first identified when the cancerous cell was observed. When cancerous cells was observed, it was found that the telomere was not shortened. It is very intact, and that is why they replicate very fast too. Then coming to the functions of the telomeres, the telomere protect the chromosome. So telomeres, they are the protect the chromosome from sticking together. The telomeres help the chromosomes from sticking together, and they separate one chromosome from another in the DNA sequence, and without telomeres the end of the chromosome would be repaired, leading to chromosome fusion and massive genomic instability. Because of the presence of this telomere, the chromosome will not stick together. Then they prevent the they protect the chromosome from nuclease activity. Nuclease are enzymes which degrade the nucleic acids or the genetic material. So this telomeres protect the new activity of nuclease. Then coming to the telomerase enzyme. What is the importance of this telomerase enzyme? It's very important. Telomere and telomerase is important. The telomerase enzyme, the telomeric sequence is maintained by this telomerase enzyme, and it is a very unique enzyme. The telomerase enzyme helps in maintaining the integrity or the tip of the telomere. So that is the function of telomerase enzyme. Telomere, tip of the chromosome; centromere, center of the chromosome. Now let's have move through the chromosome number. That is, the chromosome number is the number of chromosomes in a somatic cell, and it is designated as 2n. And in human beings, there are 23 pairs of chromosomes, and out of these 23 pairs of chromosomes, the 22 are autosomes. The 22 chromosomes are 22 are autosomes, and remaining one pair are sex chromosomes. In males, the sex chromosome will be XY, and in females, it is XX. This is the chromosome constitution of the humans. And with this, I conclude the session. Thank you. Next session will be about the giant chromosomes, heterochromatin and euchromatin. Thank you.